I am Prashant Joseph, working as an application engineer for LCMS small molecule applications development. I work at Agilent Center of Excellence Lab in Bangalore, India. In this session, we will discuss on the high sensitivity accurate trace level quantitation of nitroso drug substance related impurities or NDSRIs in various drug substances and products using LC-MS technique. Let me re-stress again on the importance of quantification of mutagenic impurities at trace levels in various drug substances and drug products. Many conventional nitrosamines and NDSRIs are recognized as mutagens and potent rodent carcinogens. These class of compounds are included in a cohort of concern because of their potential human health risks. Over the past few years, nitrosamine impurities have been detected in various drugs, leading to drug recalls. API-derived complex nitrosamines are known as nitrosamine drug substance-related impurities or NDSRIs. Multiple recalls of pharmaceutical drug products due to the presence of NDSRIs have drawn attention of both regulators and manufacturers. The story started in the year of July 2021, where the first NDSRI was identified in Varenicline drug product. The NDSRI was N-nitroso-varenicline. FDA announced the recall of varenicline drug due to the unacceptable limits of this nitrosamine impurity. In the same year, another NDSRI was identified in the ARB class of drug, irbesartan. The search continued in 2022 as well, where different other class of drug compounds such as beta blockers, thiazide diuretic, also found with nitrosated compounds such as N-nitrosopropranolol, N-nitroso-orphanadrine, and N-nitroso-hydrochlorothiazide. Soon other class of drug also identified with the presence of NDSRIs. This include anti-hypoglycemic and ACE inhibitors such as Sitagliptin and Quinapril respectively. Unlike conventional nitrosamines such as NDMA, the nitroso drug substance related impurities are relatively larger molecular weight compounds. The literature presented here includes studies on 12,000 drug molecules and their impurities screened in silico to identify NDSRI precursors. Among these molecules, 40% of the drug molecules and 29% of the drug impurities were found to potential nitrosamine precursors. Let's discuss on the formation of nitroso drug substance related impurities. NDSRIs can be formed from both secondary and tertiary amines. Secondary amines can easily undergo nitrosation in the presence of trace amounts of acid. In the case of tertiary amines, this reaction is possible due to nitrosative cleavage or dealkylation. Another way of formation of NDSRI in drug substance is reported as degradation of API. The classical example is illustrated here, which is the NDSRI NTTP formation from Sitagliptin. By considering the complexity of global pharmaceutical supply chain, manufacturers must be more diligent to protect consumers by screening their APIs and drug products for the presence of NDSRIs. In this slide, we can see that the 5 predicted carcinogenic potency categories and associated recommended allowable intake limits for these compounds. For potency category 1, the recommended allowable intake is 26.5 nanograms per day, whereas for potency category 5, it is 1500 nanograms per day. The specification limit can be calculated by dividing the allowable intake with maximum daily dose. The AI for NDSRI class 1 compound impurity is 26 nanogram per day. Say for example, if the maximum daily dose for a tablet is 100 milligram, the limit of this N-nitroso impurity in ppm was calculated as 26.5 nanograms per day divided by 100 milligrams per day, which is equivalent to 0.265 ppm. Now let's discuss on the challenges involved in the method development for the analysis of NDSRIs. Sensitivity and lower limit of quantitation are important aspects while method development. LOQ of 10% of the specification limit is generally preferred. In general, electrospray ionization is the ionization mode for these compounds, but there are exceptions where we may need to use APCI source as well. Inclusion of additives such as ammonium acetate, ammonium formate, ammonium trifluoroacetate, etc. are followed to improve the sensitivity. If the API is not stable in a diluent, NDSRI response may increase on storage. This is majorly due to the hydrolytic and pH dependent degradation and conversion of API to their respective NDSRI compounds. The diluent basified as per the need, maintain autosampler temperature, avoiding water in diluent, etc. are few of the steps to avoid degradation of API in solution. Low recovery at lower concentration levels and high recovery due to unstable API in diluent and presence of interferences are few challenges faced during recovery studies. Carryover majorly associated with the interaction of analyte with column stationary phase. This can be minimized by optimizing the gradient program. Agilent 6475 LC triple quadrupole has been proven to be analytically sensitive, robust and reliable. Vacuum shield is at the inlet of the instrument, which helps you to do the most common front-end maintenance quickly without breaking the vacuum. An octuple ion guide transmits a wide range of mass range from 5 Dalton to 3000 Dalton. The heated hyperbolic quadrupoles, since they are heated, they stay extremely clean and maintain the stability for a longer time. The hexapole collision cell is curved, where the curvature cut down the noise substantially, but also tapered to allow for refocusing of ions for very high sensitivity. The detector assembly of a 20 kilovolt high energy diode coupled to an electron multiplier horn proven to give us 6 orders of linear dynamic range. Agilent LC triple quartz have an extensive ion source portfolio that lets the user to choose the ideal ionization source for their application. The standard electrospray ionization is used for the broadest analyte ionization, which includes both small and large molecules. The modified electrospray ionization source, Agilent Jetstream, is with additional heaters to improve ion formation to achieve the ultimate sensitivity. The APCI source is used for the analysis of small molecule analytes complementary to ESI. Multimode source allow the user to simultaneously apply ESI and APCI technique within the same chromatographic run. Mass center quantitative analysis has been the premier tool for quantitative mass spectrometry analysis. There are powerful tools available in the software to efficiently review the data. User can review the peaks in a single window with compounds at a glance feature. User can also monitor the ion ratios and identify the outliers. With QuantMyWay, a user can customize the user interface to reduce the complexity and focus on the tools they use the most. Now let's discuss on few methods developed for the trace level accurate quantitation of NDSRIs. Varenicline tartrate is an active pharmaceutical ingredient found within some smoking cessation medications. In 2021, it was discovered that varenicline tartrate can form a nitroso drug substance related impurity that can potentially be carcinogenic. Therefore, the medication was voluntarily recalled to mitigate the adverse effect of administering the drug product. In response, FDA determined an acceptable daily intake level of 37 nanogram per day and released a method to analyze Varenicline and NDSRI in both Varenicline drug substance and drug product. Due to the possible carcinogenicity of the impurity, it is important to provide a sensitive and robust method to analyze the compound within the Varenicline drug product to control the amount of the impurity delivered to the patient. In this method, we used electrospray ionization source in positive mode for the ionization of analyte. Reverse phase chromatography with aqueous and organic phase containing 0.1% formic acid is employed for achieving the separation between API, impurity and the excipients from formulations. Agilent Infinity Lab Poroshel HPHC18 is used as stationary phase, which provided sharp peaks for the impurity even at low concentration levels. A diverter valve program is set to divert the high concentration API to waste and selectively quantify the NDSRI impurity. API is monitored with the help of a diode array detector set at a wavelength of 237 nanometers. Let's discuss on the sample preparation for Varenicline tablets. Using the label concentration, crush enough tablets to get a uniform sample. Take appropriate weight considering a target concentration of 0.5 milligrams per ml of Varenicline API in methanol. Vortex for 1 minute followed by 40 minutes of shaking using a shaker. Filter the supernatant solution using 0.22 micrometer PVDF membrane filter and transfer into HPLC vial for analysis. In this slide, we can see the MRM conditions optimized for the analysis of N-nitroso-varenicline. 241 is taken as a precursor ion and 211 is taken as a quantifier ion. The qualifier ion is 194. We can see a chromatogram at 1 nanogram per ml concentration, which is equivalent to 2 ppm with respect to test. As per the available US FDA method, the LOQ achieved was 0.5 nanogram per ml, which is equivalent to 1 ppm with respect to test. However, in the Agilent developed method, LOQ found is 0.25 nanogram per ml, which is equivalent to 0.5 ppm with respect to test. Also, we can see that the area response achieved at LOQ level is very consistent. With 7 injections, the percentage RST for the area response came to 7%. When we look at the recovery, which is performed at 2 formulations, one at 0.5 mg and another one at 1 mg tablet, we see that the recovery values are 104 and 110. Here the recovery is performed at a 10 nanogram per ml level because there was already a content in the sample. Therefore, we could not go to the recovery study at LOQ level. Dabigatran is an anticoagulant used to treat and prevent blood clots and to prevent stroke in people with atrial fibrillation. Analysis of N-nitroso-dabigatran in dabigatran ethoxylate mesylate is one of the most demanding applications in the NDSRI arena. In March 2023, from the US market, a voluntary recall happened due to the presence of N-nitroso-dabigatran impurity. Agilent 6495 LC triple quad is used for the method development for the analysis of this impurity. Agilent Poroshel HPH column was used for the chromatographic separation between the API and the impurity. Almost 3 minute separation was achieved between API and N-nitroso-dabigatran in the developed chromatographic method. We use 1 millimolar ammonium trifluoroacetate to improve the sensitivity of this impurity in ESI positive mode of ionization. Dabigatran ethoxylate mesylate showed the absorbance in UV at 273 nanometer. Let's understand the sample preparation for Dabigatran ethoxylate mesylate API. Here it is important to know the storage conditions for this API. API samples are stored with a desiccant at a temperature between 2 to 8 degrees Celsius in dark. So this is to avoid any moisture and also photodegradation. API samples of 20 milligrams were dissolved in 4 ml of diluent in a volumetric flask and then vortexed at 2000 rpm for 10 minutes. So here we basify the diluent in order to stabilize the API in solution. Filter the supernatant solution using a 0.22 micrometer PVDF membrane filter and transfer to HPLC vial for analysis. API sample is monitored by UV at 360 nanometers. High concentration API is diverted to waste and selectively send the impurity to MS for accurate low-level quantitation. We found that the blank is completely free from all the interferences. An LOD of 6 picogram per ml and LOQ of 10 picogram per ml absolute were achieved with this method. The response was linear with a concentration from 10 picogram per ml to 1000 picogram per ml with a regression coefficient of 0.9994. Highly reproducible responses were achieved at a standard at specification limit and for the recovery samples at LOQ and at specification. The percentage RST for area response of 3 injections each from 2 different preparations found to be less than 3%. The recovery at different levels including the LOQ found to be between 95 to 110%. To understand the concerning contamination with nitrosamines on Sitagliptin Metformin drug product, an analytical method is developed using Agilent 6475 LC triple quad for the determination of N-nitrosotriazole pyrazine. This method describes the procedure for the MRN-based low-level quantitation of N-nitrosositagliptin impurity using APCI source. In the developed chromatography using a poroshel phenylhexyl column, metformin was eluting at 1.7 minutes. A 25 minutes gradient was employed to achieve the separation between the APIs and the impurity. The sitagliptin tablet control sample was showing a response at 2 places in chromatogram. One is at 1.5 minutes and another at 11.96 minutes. The impurity showed an RT of approximately 10.5 minutes. Hence, the diverter program is set for diverting both metformin and sitagliptin APIs into waste. There are literatures on method for the analysis of sitagliptin developed in ESI positive mode. However, we found that APCI was giving better sensitivity and selectivity for the analysis of this impurity. Let's discuss on the sample preparation for sitagliptin and metformin combination drug product. First, accurately weigh 15 mg sitagliptin equivalent tablet crush powder and transfer to a 15 ml centrifuge tube. Add 5 ml of diluent, vortex the sample for a minute, followed by 40 minutes of shaking using a rotospin shaker. Post-effective extraction, centrifuge the samples at 4000 rpm, maintaining 10 degrees Celsius for 20 minutes. Filter the supernatant solution using a 0.22 micrometer PVDF filter. This filtered solution is used for LCMS analysis. Linearity from 0.04 nanogram per ml to 1.3 nanogram per ml is plotted and found to be linear. The regression coefficient obtained was above 0.99. LOQ of the NDSRI impurity is 0.08 ppb, which is equivalent to 0.027 ppm with respect to a test concentration of 3 milligrams per ml. At LOQ level of 0.08 nanogram per ml, the area response found to be consistent with a percentage RST of 2.6% shows the reproducibility of instrumental analysis. Recovery study conducted at spec level with 3 different preparations, and recovery of NDSRI N-nitrosositagliptin in tablet formulation was found to be 116%, 109% and 114% respectively. There was a news on recall of propranolol tablets of various strengths by Pfizer Canada due to the presence of N-nitrosopropranolol. There is a need to develop a highly sensitive LC-MS method for the quantification of this NDSRI impurity in API and tablets. Here we developed this method and established all the critical method performance parameters, including the limit of detection, limit of quantification, specificity, linearity, reproducibility, and recovery. Agilent Infinity Lab pursued diphenyl XRS column with an 18 minutes gradient is employed for the chromatographic separation between API, NDSRI impurity and N-formyl impurity. Since there is a little information available regarding the limits of N-nitrosopropranolol, the method was developed with a widely accepted limit of 0.03 ppm in relation to propranolol sample concentration. However, in this method we achieved a much lower limit of quantification of 0.005 ppm with respect to the propranolol sample concentration that corresponds to an absolute concentration of 25 picogram per ml of N-nitrosopropranolol impurity. Sample preparation was optimized in terms of extraction solvent and time and achieved the desired recovery for N-nitrosopropranolol in propranolol API and tablets. There are 2 different sample preparation methods adopted here. In this slide, we discuss on the sample preparation for the extraction of N-nitrosopropranolol in propranolol API sample. Accurately weigh 25 mg of the drug API into a 15 ml centrifuge tube. Add 5 ml of the sample diluent, which makes a test concentration of 5 mg per ml. Vortex the solution for 1 minute, followed by 15 minutes of sonegation so that API is completely soluble. Transfer the solution to an HPLC vial and load it to LC-MS for analysis. Now, let's discuss on the sample preparation for 40 mg tablet formulation. Using the label concentration, crush enough tablets to obtain at least 3 times the API target weight. Find the average weight per tablet and accurately weigh the equivalent of 25 mg API. Transfer to a 15 ml centrifuge tube and add 5 ml of the diluent, which makes the API final sample concentration of 5 mg per ml. Vortex for 1 minute, followed by 40 minutes of shaking using a shaker. After extraction, centrifuge the samples at 4500 rpm for 15 minutes. Filter the supernatant solution using a 0.22 micrometer PVDF membrane filter and transfer to an HPLC vial for LC-MS analysis. A 4-minute separation is achieved between API and the N-nitrosopropranolol impurity. The LOQ achieved was 0.005 ppm with respect to a test concentration of 5 mg per ml. The calibration curve for N-nitrosopropranolol demonstrated an accuracy rate within 15% of the expected concentration. The recovery experiment was performed in duplicate for both propranolol API and 40 mg tablets. In tablet, the recovery experiment was performed at a higher concentration due to the presence of N-nitrosopropranolol present in the samples. Data found highly reproducible across all levels, especially at LOQ, exhibited CVs less than 5%. Formic acid or formaldehyde is frequently responsible for the formation of N-formyl impurities from drug substances that contains primary or secondary amino moieties. To establish the specificity, need to establish the separation of N-formyl impurity from the NDSRI. Here we give a representative chromatogram showing the separation of propranolol formyl impurity and N-nitrosopropranolol at 0.6 nanogram per ml. This separation demonstrates the specificity of this developed method. Valsartan is a medication used to treat high blood pressure and heart failure. Nitroso-N-D-valeryl valsartan methyl ester is a chemical impurity that is commonly found in valsartan. This impurity is a mixture of isomers with the exact composition varying depending on the manufacturing process. This impurity comes in both ESI positive mode as well as in ESI negative mode. However, we finalized the method with ESI positive mode using Agilent Jetstream source. An Agilent Infinity Lab Poroshal phenyl hexyl column is employed to achieve the separation between the placebo and the impurity. 1 millimolar ammonium trifluoroacetate is considerably improved the sensitivity of this impurity. Let's discuss the sample preparation for placebo. Accurately weigh 25 mg of placebo into a 15 ml centrifuge tube and add 5 ml of sample diluent. So this makes a test concentration of 5 mg per ml. Vortex the solution for a minute followed by 15 minutes of sonation. Transfer the solution to HPLC vials and load into LCMS for analysis. An LOQ of 7.5 picogram per ml absolute equivalent to 0.0015 ppm is achieved in placebo. The calibration curve is from 3 picogram per ml to 30 picogram per ml, found to be linear with a regression coefficient of 0.9982. The accuracy of different calibration levels used in the linearity were within 80 to 120%. The intraday reproducibility was measured for 3 consecutive days, and the percentage CV for area response of 18 injections were found to be 6.1%. The average recovery at LOQ level studied with 6 different preparations and found to be 91.6%. These are some of the ready methods for the quantitation of NDSRIs in various drug substances and products. In all these methods, the instrument sensitivity is far better than the required regulatory limits. The result reproducibility is exceptional with very low percentage of RSTs at LOQ levels. Agilent AJS and APCA sources are very stable ionization sources, enhance the sensitivity and reduce contamination. The recovery is achieved for all NDSRIs in various drug substances and drug products were well within the regulatory acceptance criteria. With that, I conclude my presentation. Thank you very much for your kind attention.